My name is Yang Ken. I'm a professor of cancer cell biology from Shanghai Tech University in Shanghai. I'm also a scientific advisor to Oncology and Immunology Unit of WuXi AppTec. So today, I'm going to discuss two topics. So first, I will present our latest research on how to use CRISPR library screening to discover novel oncology and oncoimmunology targets. I will then discuss some technical considerations for conducting successful CRISPR screening in vitro and in vivo. So we all agree cancer is a genetic disease. So somatic mutations accumulate in adult cells over time to drive intrinsic cell proliferation, as well as allowing the cancer cells to overcome environmental constraints. So systematic efforts have already been ongoing to identify genes essential for viability across hundreds of cancer cell lines, which are growing petri dishes. So we routinely these days look up our favorite genes in that map provided by Project Achilles, how our favorite genes behave in vitro. However, the essentiality of genes often change when the cells are growing under different conditions. For instance, we very likely will pull out different sets of hits when we grow the cells as a 3D spheroids as compared to the 2D cultures, or when we culture the cells with therapeutic compounds or T cells in vitro systems, or when we grow the cells as a xenograft challenged with or without immunotherapeutic therapies. So in the next few slides, I'm going to discuss several examples how we use CRISPR library screening to interrogate drug-resistant mechanisms as well as identify synthetic lethal targets. So tumors inevitably develop resistance to targeted therapies. So when we have effective therapeutic compounds, we want to understand we could use CRISPR library screening to identify the pathways controlling the resistance of the cancer cells to the therapeutic compounds. So in this case, we introduced a whole genome library to a pomalidomide-sensitive multiple myeloma cell lines, MMS cell line. And then we challenged these cell lines with the library with the compound pomalidomide. And we harvested cells at different time points, and in the end, analyze the change of the abundance of different guides after the cells are challenged with the pomalidomide. So cerebrum is a direct target of pomalidomide. As you can appreciate, that all the targets against cerebrum could be identified after the cells are exposed to the compound. Surprisingly, we also pull out almost all of the components targeting the COP9 signosome. So, and we eventually figured out that the COP9 signosome regulate the activity of a different E3 ubiquitin ligase, which regulates the turnover of our cerebrum. So very often, our compound are not as effective as we would wish. So with the limited efficacy, we were very interested in identify the prognostic marker for most susceptible patients. And we want to identify synthetic lethal targets for future combination therapies. So in this case, we introduced a whole genome library to the pancreatic cancer cell lines. and then challenged this library with a V1, a G2M cell cycle checkpoint inhibitor at a lethal dose 20, so meaning only 20% of the killing at time of the treatment. And then we harvest the cells at different time points. I just want to mention that the initial coverage is between 500 to 1,000, since this is a negative selection of targets. So by Empiricon PCR and next generation sequencing, we apply the criteria shown on the bottom right corner. When we use a 1.5-fold change as a cutoff, and right FDR, and at least 3 guides as a selection marker, we could identify only 17 genes out of this whole genome library that, when inactivated, cause synthetic lethality with this cell cycle checkpoint inhibitor. So amazingly, almost all of these encode regulators of the cell cycle, particularly those regulating cytokinesis and actin filament regulation. So we are also surprised to identify 3 out of 6 enzymes regulating the heme biosynthesis, suggesting a heme-regulated pathway in regulating the drug sensitivity in this particular screening. We could apply selection pressure more than just the small molecule chemicals. According to this cancer immunoediting model, the cancer cells have to overcome the restriction of host immune systems to become a tumor. So they achieve this goal by accumulating mutations to allow the cells to escape immune elimination. So how can we identify such mutations? So in the next few slides, I'm going to show in details how we perform in vivo CRISPR screening to identify mutations that sensitize cancer cells to immune checkpoint blockers in xenograft models. So for in vivo CRISPR screening, we cannot use whole genome libraries because we have to have enough or sufficient guide coverage. We typically start with at least 500 times coverage of each guides in the initial samples. So in this study, we generated a customized CRISPR library with about 2,000 guides targeting about 300 genes that have been reported to be highly mutated in cancer genomes. We introduced this library to a mouse colorectal cancer cell lines, MC38 ova, expressing low levels of ova peptide. Then we implanted these cells into a syngenic B6 mice. When the tumors start to grow, we treated these animals with anti-mouse PD-1 antibody or mouse IgG. In the end, we harvested the tumors when the control tumors reached about 1,000 cubic millimeter, and this model is very sensitive to PD-1 immune checkpoint blockers because the PD-1 antibody administered every 2 days can effectively inhibit the tumor growth. So these tumors were harvested. We extracted genomic DNA, amplified the empiricons, and subjected them for next generation sequencing. So we could identify these many guides against these genes to be deleted in the immune checkpoint treated animals as compared to the control animals. So these genes encode PBRN1, array2, KMT2D, and KDM6A. So these, interestingly, these PBRN1, array2, and BRD7 forms a complex called PBF Sniff Switch complex, which regulates chromatin accessibility. The components of this complex have been identified to suppress T-cell mediated cytotoxicity in a cancer cell T-cell co-culture screening published about 2 years ago. PBRN1 of the complex have also been identified to be highly mutated in renal cancer patients that show great response to the immune checkpoint therapies in patients. So we hypothesized that like these PBF Sniff Switch complex, KMT2D and KDM6A might also function similarly. So KMT2D and KD6A are frequently mutated in human cancers and a rare genetic disease called Kapoki syndrome. KMT2D in particular is a major histone H3K4 monomethyltransferase required for enhancer activation and cell type specific gene expression. So to validate the KMT2D in this as a sensitizer to PD-1 immunotherapy, we performed this in vivo competition assay. So briefly, we knocked out KMT2D in M-cherry expressing cancer cells and mixed them with EGFP expression control cells. Then we implanted this mixture into syngeneic hosts and treated these hosts with anti-mouse PD-1 or anti-mouse PD-L1. In the end, we harvested the tumors, dissociated the cells, and ran FACS analysis for the composition of EGFP and M-cherry expressing cells. As expected, loss of array2, one of the components of the PBF complex, sensitize the cells to PD-1 or PD-L1 induced killing. Loss of KMT2D also sensitized to the checkpoint blocker, but at the cost of the increased growth of the KMT2D knockout cells. So in another word, the growth advantage obtained by KMT2D knockout in vivo is offset by the PD-1 immune checkpoint in this model system. So this is a working hypothesis that we are still working on right now. So I have discussed how we use CRISPR library screening to identify synthetic lethal targets, either to chemical inhibitors or immune checkpoint blockers, as well as to investigate the drug-resistant mechanisms. So for the rest of the time, I'm going to discuss some technical considerations for successful CRISPR library screening. So in WuXi, we typically follow these screen flowchart to perform our screening. We can start with a design of a focused guide RNA library to construct the library, produce the virus, and perform the screening in-house, as well as the NGS and final data analysis. For focused, for customized library design, we derive the sequence against the genes of interest from public databases, as well as the historical data from our own screening. I would like to mention we have performed hundreds, if not thousands, of CRISPR screening in vitro and in vivo. We have accumulated a lot of in-house information about the performance of individual guides in our screening. And we take into consideration this information in our CRISPR library design. We also use non-targeting guides and non-essential genes in our library design. We use both all-in-one vector system and two-vector systems for our screening. And in our hands, the targeting efficiency by these two different systems are not significantly different in most of the genes we have studied. So the choice of one vector system or two vector systems in the end very much depends on the experimental design, the timeline, as well as the cost associated. As I said, we have done many screening, and some cell lines are very easy to screen against, and some are simply resistant to be screened against. So we have a huge list of cell lines we have performed successful screening against. If you don't know which cell line to start with, we would recommend you could look up our screening list and design your screen. Balanced distribution and sufficient coverage of the guides in the library is very important for eventual successful interpretation of the data. So this is a QC analysis sample from the in vivo CRISPR screening I have just presented. As you can appreciate, for this screening, the read counts are evenly distributed across all the samples from the start to the end. The Gini index reveal very little unbalance of the guides in these samples. And the non-targeting guide RNA sequences are well represented in all of the samples. We use read counts to correlate all the samples and exclude those outliers from final analysis. We also use the percentage of zero counts in all the samples to demonstrate the good representation of the guide in the samples we're going to analyze in the end. So finally, we use HR and different versions of MAGIC for statistic analysis of the guides and genes targeted by these guides. We also use the non-essential genes and positive controls, which basically is based on published data to fine-tune our initial unbiased analysis of the targets. I would like to mention that the CRISPR screening is a start of the efforts to identify targets. Once a target is identified, the team in WuXi could engineer cell lines or animal models to investigate the biology of the targets so that we can determine if the targets can be therapeutically developed in the end. So this is very much for my presentation. Again, I want to emphasize that all of the screening has been done by a talented team in WuXi AppTech, led by Dr. Chunshen Ji. The mechanistic studies and some experimental design were performed by graduate students and postdocs in my lab in Shanghai Tech University. So if you are interested in exploring CRISPR screening, for your drug target discovery efforts, please feel free to contact Dr. Declan Wine, who's based in Boston, and who's also Vice President of the BD team of WuXi AppTec. So I will stop here, and if you have any questions, please feel free to direct to Declan. Thank you.